Okay, so yesterday we talked about regulation of gene expression in prokaryotes, and not that that's a simple process by any means, but prokaryotic cells are less variable and less complex than eukaryotic cells, and so a lot of the phenotypes in prokaryotic cells are fairly simple. So the gene expression doesn't levels of gene expression don't change all that much outside of metabolism and outside of how replication takes place. In eukaryotic cells, even in single-celled eukaryotic cells, you have a lot more variability in terms of what is expressed, how much is expressed, and because of that, eukaryotic gene regulation is more complex than prokaryotic gene regulation. And so a couple of images here. Here are images of various yeast cells that are expressing different genes. And using fluorescent labels, you can see different activity going on in these different cells based on which genes are being expressed and at what level. Over here, you've got a zebrafish embryo, and you have green fluorescent protein here that is being expressed whenever the Hox gene PAX1 is being expressed, and PAX1 is a master control gene that helps to develop eyes and eye structure and neurons. And so you can see wherever PAX1, that master control gene, is being expressed, it leads to the expression of this green fluorescent protein. This is something that was added to the zebrafish, is not natural there. So you could see where that box is actually where that Hox gene, the homeobox gene PAX1, is being expressed. Over here, this is an earthworm, and it's showing you the expression in neurons. And so you can see here there's a body of a neuron and then a long axon stretching out this way. And so again, this is using green fluorescent protein, and which is triggered by the expression of another gene, expression of genes in neurons. So we can use this to map various structures within an organism. And all of these images just to kind of show you how we study gene regulation and how we take that information to figure out exactly what's going on, and then from there to go in and figure out how all of these levels of expression are regulated. And so in eukaryotic cells, there are three main places in which you can regulate gene expression. The first one is transcriptional regulation, and this is what we're going to spend most of our time talking about because the best bet for the cell energetically is to not invest in something that you're not going to use. And so if you're going to regulate it at the best place for energetics, it's going to require, not transcribing it in the first place, or transcribing less or more of it as necessary. Then we can regulate mRNA. We talked about when we talked about transcription and translation and gene expression last week, we talked about RNA modification. And so in that process of maturing your mRNA, we have a high level of control. Many of you talked about that on your quiz in terms of addressing postulate as to why, the cell actually makes pre-mRNA that has to be processed to be mRNA. And one of those reasons is to allow a place to shut down gene expression if necessary, or to regulate it. And then the other one, and this is, as a last step, protein regulation. And this is going to involve mainly marking proteins for degradation. And you do that with putting a ubiquinone marker on it. You call that ubiquitation, or even the simulation, the small ubiquitin like modifier. Again, those are marking proteins for degradation. And I mentioned homeobox genes, specifically PAX1, in the previous slide. And so what these homeobox genes are, master control genes. They are genes that produce protein products that turn on the transcription of other genes. And so if those master control genes are not turned on, if a specific master control gene is not turned on, it will not turn on the transcription of whatever the genes are that function under that. And so these master control genes are basically at almost the highest level of gene expression. And so when they are transcribed, they turn on a suite of other genes. And this is what you find in multicellular organisms, where, let's say you're thinking about a neuron versus a liver cell. And so all cells in our body, let's use a human neuron and a human liver cell, all cells in our body have what are called housekeeping genes. And so if you took any cell from the human body and you measured the amount of gene expression going on in that cell, what you're going to find is that there are certain what are called housekeeping genes that are necessary just to keep the cell alive, that are producing the proteins of glycolysis, that are producing proteins for metabolizing other materials, that are these housekeeping genes. And so every cell is going to express those. But then a neuron is going to express a different suite of genes than a liver cell is, and that is controlled in a lot of ways by which Hox gene is active, and then therefore which suite of genes get turned on and allow the transcription. And so as we talk about transcription regulation, I want you to think about it in the context of turning on a master control gene that triggers the transcription of other genes. So think about it in that context. All right, so this is the main way in which it seems that cells regulate transcription and regulate the amount and regulate whether or not transcription even happens. And so you can see in both of these examples with histone acetylases and with nucleosome remodeling proteins, in this case we have condensed chromatin. We would call this heterochromatin. Chromatin that's condensed, and you cannot access the promoter. The transcription factors can't access this promoter. They can't ensure that RNA polymerase binds at the correct place and transcription happens. And so whatever genes are packed into chromatin like this, you can't express those. And so this is the main way in which a cell regulates and controls the amount of transcription in most of its genes, is by packing the chromatin as densely as possible, and so you can't access that promoter. And there are two main ways in which you can form what you would call euchromatin, where you can access those promoters. One is by acetylating the histones. And so you see all of these histones here. There are 6 of them that have been acetylated. And so when you add that acetyl group to the histones, it doesn't allow those histones to be packed as tightly together. Therefore, it's a little bit looser, and now the machinery, transcription factors, can access the promoter, can ensure that RNA polymerase binds to the correct site, and can ensure that transcription can progress. And so this is one way. This histone acetylases, this is more long-term. Now, that's not to say that you can't deacetylate these, because you absolutely can, and we even talk about that in a little bit, that you can shut down the transcription of a particular gene by deacetylating the histones where that gene is located in the chromosome. But this is more long term, and so if you were to look at a liver cell and you know where the genes are that code for proteins that are active in liver cells, let's say alcohol dehydrogenase, the enzyme that metabolizes ethanol. You know where that's located in the genome, and you went and looked on the chromosome where that gene is, you're going to find acetyl groups there forming euchromatin so that the machinery can access that and transcription can happen. This form here, this is also a way in which you can convert from heterochromatin to euchromatin and access the promoter and access that portion that needs to be transcribed. The difference is these remodeling proteins that will unwind it a little bit, they don't keep the histones from being packed tightly together. What they do is they disrupt the way in which DNA is wound around these histones. You can see it's getting a little bit looser, and you're able to access that better. And then this is more short term. And so say you have, a gene that the cell only turns on when it's induced by a particular hormone that it binds at a receptor. And so this would probably be the way that the cell would respond to that, that a signal transduction event would happen in response to that hormone binding, and ultimately it would lead to some protein entering into the nucleus, and that protein would then cause a bit of a remodeling to make it to where the transcription factors can access this promoter, and the transcription of that gene can progress, and then as soon as those proteins leave, it returns. But in this situation over here, you would actually have to deacetylate those in order to get those histones to pack tightly again and recondense that chromosome. So again, this is more long-term activation. This is more short-term activation in response to some kind of a cell signal. And here, we talked about this when we talked about transcription, enhancer sequences, and these could be either cis, which we talked about on Monday, means immediately next to the gene, or they could be trans. And really what we're finding is that these enhancers could probably be anywhere in the genome, even on a separate chromosome, and inside of the nucleus, the cell could reorganize the chromosomes in such a way that it can bring any part of the genome, even on a separate chromosome, in close enough proximity to whatever this gene is, to increase the rate of transcription. And that actually brings up a good point about this previous information. It seems to be that the packaging of one chromosome can influence the transcription of another chromosome. So let's say, for instance, on chromosome 1, you had these acetylated histones, and you have euchromatin, you have this loose chromatin that you can access with your transcription machinery. And then chromosome 2 comes in close proximity to this. Chromosome 2 can actually be influenced by the three-dimensional structure of chromosome 1 to loosen itself and to allow the transcription machinery access to genes. And then the opposite is true as well. Let's say chromosome 1 was structured like this, where the histones are packed really tightly together. You have heterochromatin, and you can't access the genes with the transcription machinery. Chromosome 2 coming in close proximity to that could actually shut down the transcription of genes on chromosome 2. And so there's a lot going on inside of the nucleus that we're still learning about, and we're still figuring out how much control the cells actually have over transcription, the rate of transcription, and whether or not it even progresses. So going back to here, again, these enhancers can be cis, they can be right next to the gene, or they can be trans, located probably anywhere in the genome. And enhancers function in two main ways. They're enhancers. They increase the rate of transcription, and they do that primarily by allowing activators to bind to them, and the activators recruit these co-activators called mediators that make it really easy for RNA polymerase to bind to this initiator sequence and to begin transcription. And so it's not that RNA polymerase wouldn't get there on its own. As long as these transcription factors bind the base promoter, the Tata box in eukaryotic genomes, as long as those transcription factors bind there, RNA polymerase is going to bind to the initiator sequence, and initiation of transcription is going to start. But these activators, binding co-activator, the mediator, increases the rate at which RNA polymerase binds to that. Keeping in mind that you can have multiple RNA polymerases transcribing this gene at any given time. One could be really far downstream towards the end of the transcription process, whereas another one's in the middle, another one's a little bit, further upstream in the middle, and one's at the initiator sequence. So you can have multiple RNA polymerases transcribing any one gene at any given time, and these activators binding the coactivators, the mediators, increase the rate at which you recruit RNA polymerase, therefore making it to where you're producing more mRNA at a much faster rate. And so this is a big way in which cells control gene expression, is by activating that expression, by allowing activators to bind to the enhancers and recruit RNA polymerase. So these are for genes that are already on euchromatin. They're already accessible with the machinery, with the transcription machinery. You're just enhancing the rate at which that transcription happens. You're ensuring that you're making more mRNA, and these are coding RNAs, the pre-mRNA. And so here are some of the activators. We talked about this last week when we talked about transcription, and we talked about these different domains, the helix turn helix, the zinc finger, the leucine zipper, and these are all proteins that bind as activators. You can see that down here, bind as activators at the enhancer sites. In these examples here, these are cis sites close to the gene itself. And these, again, these are not binding transcription factors. These are not ensuring that RNA polymerase binds to the correct place. That's going to happen. These are recruiting RNA polymerase and making it happen more rapidly. You might think of them as a catalyst, even though they're not, helping to overcome the activation energy, but they are increasing the rate of the reaction. Most of these are active as dimers, either homodimers, where you would have two proteins with the helix turn helix domain or motif, or a heterodimer, where you would say have one protein with the helix turn helix and one protein with the zinc finger. Regardless of these domains or these motifs, what you tend to find is that each of these have a alpha helix that will get in and will interact with one of the strands of the DNA molecule, helping to actually pull the two strands of DNA apart. And you can see that illustrated here with the leucine zipper, helping to pull the two strands of DNA apart, making it easier for the transcription machinery to access these sites. And so again, it's not, they're not transcription they are transcription factors, but they're not the transcription factors that ensure RNA polymerase binds to the correct place. These are activators that make it to where all of that happens more quickly. And again, all of this is in the area of regulating gene expression by enhancing the rate of transcription for genes that are already being transcribed. So the previous material that we talked about of acetylating it, that it makes it to where transcription is possible. The enhancers make it to where that transcription happens more rapidly. And here's a way in which you can actually regulate the expression in a place of the chromosome that is euchromatin, that is already opened up enough that the transcription machinery can access. You can actually slow this down. You can repress it, not completely eliminate the transcription. You can repress it by having a repressor protein actually block activators from binding to the enhancer. And so these activators are, if we go back here, let's remove the repressor, activators binding here at the enhancer site, recruiting this coactivator, the mediator, that's pulling RNA polymerase to the correct place, helping that happen more rapidly. And then a repressor will come in, will bind to that site, and so it won't recruit the mediator, it won't help transcription factors bind to the promoter, the Tata box, and it won't pull RNA polymerase as well. And so that's still going to happen, again, unless you condense the chromosome so tightly to form heterochromatin that you cannot access it with the machinery. So this is repressors, this is slowing the rate of transcription. The expression is still happening, but it's happening at a lower rate. And so already we've seen that cells in the three-dimensional space of the nucleus can condense chromosomes to completely shut down transcription. They can move chromosomes over close to a portion of a chromosome that's completely shut down and will be for the life of the cell. They can move a chromosome over there temporarily to influence that chromosome to shut it down for a short period of time and then move that chromosome somewhere else, allowing transcription to happen. Or underneath that, cells regulating the rate of transcription where transcription is already happening by using activators to recruit RNA polymerase more rapidly, or by using repressors to keep it to where activators can't do that and the transcription factors have a harder time binding. And then repressors can actually recruit histone deacetylase. And this is what I mentioned on our first slide, that acetylating chromatin or chromosomes to make euchromatin, that's long-term activation, but it can be reversed by this enzyme called histone deacetylase, which remove those acetyl groups, allow the histones to condense more and shut down the transcription. And so some proteins can actually act as an activator or a repressor depending on which chromosome they're acting on and probably the overall three-dimensional structure of what's going on inside of the nucleus. All right, so that takes us past our discussion on modifying transcription and regulating transcription. And again, remember, that's the main way the cell wants to do it, because if you're really going to want to shut down the transcription, you don't want to make the mRNA and then regulate it from there. You want to just keep from investing energy in making something you're not going to use. But many of you on your quiz, when I asked you to postulate why cells make pre-mRNA, you mentioned it gives them an added layer of control, and that's absolutely correct. Because the cell in making pre-mRNA, it basically has a way in which, let's say you had the cell binds some hormone and you get that signal transduction. It codes for some protein, a regulatory protein that will go in, will modify the structure of the chromatin, will loosen that DNA up around the histones. And so you'll start transcribing, and you'll transcribe a pre-mRNA, but then whatever those conditions were are gone now. That hormone is no longer present, and now the cell needs to stop investing energy in something it doesn't need anymore. And so now you can degrade that pre-mRNA. You can take that pre-mRNA and never fully process it and degrade it and recycle those nucleotides to use with other mRNAs. We also talked about alternative splicing in our RNA modification or in our RNA maturation. And you see this especially true in different cell types, and I'll show you that, I think, in the next slide, where in different cell types you have the same gene being transcribed, but it's spliced one way in one cell line and a different way in a different cell line to produce two completely different proteins that help the cells function as two completely different cells. And then you can also control the rate at which and how these mRNAs are translated. So you can splice these pre-mRNAs in different ways. You could degrade them and never allow them to fully mature and fully be modified into mature mRNA for translation, or you can modify the rate at which those mRNAs are translated. These are all levels of control within eukaryotic cells. And so here's the alternative splicing. And here you can see in cells functioning in the thyroid, you have the same gene, the same cistron with 1, 2, 3, 4, 5 different exons. And so in thyroid cells, you cut out these interim introns, which actually function as introns in all the various cell types. And then you also cut out everything after exon 4. Whereas in neuronal cells, you cut out this section, so something that was exon 4 for here actually becomes an intron in neuronal cells, and so you get two completely different mature mRNAs. They both have the cap and the poly A tail. They've been polyadenylated and capped, so they've got the other two parts of the mRNA processing. And then they are going to be translated in completely different ways because their sequence is completely different. And so you get two completely different polypeptides that function in completely different ways. All right. So on top of that, so we've talked about how it controls the rate, how the cell controls the rate of transcription, how the cell processes pre-mRNA. We also need to talk about a whole series of non-coding RNAs. So only about 1.5% of the human genome is translated into proteins. We've now modified that, and it looks like even less than 1% of the human genome that actually gets translated into proteins. But coding RNA, mRNA, is not the only type of functional RNA. In fact, we have most of the genome, 90% or more. There was a paper published in 2012, and there's been subsequent work there that basically demonstrates that there is no portion on the human genome that is more than 8,000 bases away from a site that is actively transcribed. And so most of the genome, greater than 90% of it, is transcribed but never translated. And so it becomes part of protein or part of RNAs that are not coding RNAs, microRNAs, small nuclear RNAs, and a whole suite of other types of RNA that actually regulate which genes get transcribed. And so you can see this 10,000 plus. This is an old number. It's now 25,000 different microRNAs that we've described and shown how they regulate the expression of genes, how they regulate transcription. And so you don't just have proteins regulating transcription. Would it be histones or activators or transcription factors? But you have whole suites of RNAs that get transcribed and regulate the expression of genes. And so any particular gene can be influenced by many different microRNAs. And so this whole idea of pseudogenes, which was really popular in the latter part of the 20th century, as an argument against design, because you have huge sections of the human genome that look like they're just leftover remnants of our evolutionary history. Now what we're finding is these pseudogenes are really genes that are being transcribed into various types of RNA that we haven't fully figured out their function yet. And so this is just a lot. Basically, the take-home message here is most of the genome in our cells, most of the human genome, is being transcribed into RNAs that regulate the expression of the coding RNAs. And so most of the genome is playing a regulatory role and not a coding role. And so this adds a great deal of complexity to what's going on in our genome. It's not just information storage. It's not just gene expression of taking those blueprints and building materials with it, but it's a regulatory mechanism as well, built within the information itself. It are the machinery or the codes, the blueprints to make machinery that ensure that everything happens within the cell in an extremely controlled manner. And so here are some of the, showing you the small RNA. This is small interfering RNA, siRNA, only 19 to 20 base pairs, an incredibly small RNA. And many of these are double-stranded. And what they do is they function within the cytosol to regulate the translation, to inhibit translation, or to amplify the rate of translation, or to degrade mRNA, coding RNA. And all of these, form these complexes. You see this RNA-induced silencing complex is what this is, that function in various ways to regulate the gene expression even outside in the cytosol, outside of the nucleus. Once it's once the cell has produced mature mRNA, it doesn't lose the ability to regulate expression. It still maintains the ability to regulate expression even once the mRNA has left the nucleus. And it does this in one way by these small interfering RNAs building this RNA-induced silencing complex that can regulate the expression, either enhancing it in the case of amplification or repressing it in the case of inhibiting translation or degrading the mRNA to allow it to be recycled and no longer translated. And then the last way in which these eukaryotic cells control gene expression is by, once the polypeptide's already made, it's already folded correctly, it's a fully functioning protein. Those proteins can be turned on or off by phosphorylation. You have enzymes called kinases that phosphorylate other proteins. And so some proteins become active when you phosphorylate them, or they become deactivated when you phosphorylate them. But it's basically function as an on-off switch, taking a protein and maybe turning it off for a time and then turning it back on later. You find this a lot with proteins, with enzymes that function in metabolism. Say it's a protein that functions in the metabolism of a specific sugar, say fructose instead of glucose or galactose or maltose or mannose. And that protein, you really only want it to be active when that sugar, whenever that material is present. And so a lot of times the enzyme for regulating that or metabolizing that sugar will be turned on or off by phosphorylating it or dephosphorylating it. And then another one is just to target these proteins for degradation in a structure called the proteasome. And so the way you do that is ubiquitinate. Ubiquitinate a protein. You mark it with a ubiquitin tag, and you can see that happening here. Here's the ubiquitin tag. This protein's been marked with that tag, and then now it's going to be destined for degradation. And so actually here's the protein that's going to be degraded. So here's ubiquitin put on this protein to then get transferred over to this protein that now has this ubiquitin tag. And then once that ubiquitin tag is placed on it, it will be shuttled over to the proteasome and be marked for degradation, for destruction. And so this is a way in which the cell could shut off the expression of a particular protein long after that protein's been made, as a way of protecting itself, say shutting off a particular function or slowing down a particular function. And so all of this, again, I told you I wanted you to think about this in the context of these master control genes, these Hox genes in multicellular eukaryotic organisms. We as vertebrates have an enormous number of these Hox genes. And so the first level, or the best level to control, is to control the rate of transcription. And that level is controlled a great deal by these Hox genes. And then after that, if you're not going to shut down the transcription of it, the cell still maintains control over expression, more in how much rather than whether or not it happens at all, by either regulating the rate of transcription with activators or repressors, or regulating the rate of mRNA production by modifying pre-mRNA, or just modifying the protein itself. So the cell maintains levels of control over regulation at each level of gene expression. And where the Hox genes function is really at that first level, at whether transcription happens at all. And so that is controlled a great deal by these Hox genes, these master control genes that ensure, even early in development, certain structures develop in certain places and not in others.